Good afternoon to all the NEET aspirants 2026. This is your Prasanna Ma'am from NEET World, and today we are here to discuss about the most important topics of respiration and plants. Let us exercise the most difficult questions of TCA cycle, link reaction and anaerobic respiration, be it lactate fermentation or alcoholic fermentation. Before getting into the questionnaire session, let me give you a brief introduction about NEET World special programs. Here we have NEET World's 2 steps to medicine for 11th standard students and NEET World's 1 step to medicine for 12th standard students and NEET World's power step to medicine for repeaters. All the 3 courses are just for the minimal fee, which is 3333 per month, wherein you can access complete courses, be it offline or online, with complete study material packages and 2 master revision strokes with the same fee per month. To know more about these courses, you can call us on to the given number or you can directly visit our branch, which is in Narayanguda, Hyderabad. So, let us get started with the questionnaire sessions. Today we are going to exercise difficult questions beta. This is very important session. Don't miss it at all. So, if you have to look into this flow chart, what does A, B and C depict in the given pathways he is asking you. So, this is connecting with what? Glycolysis, lactic acid fermentation and alcoholic fermentation. And here how the energy is being floated is given in the options. We have to choose accordingly. So, here if you have to see glyceraldehyde 3-phosphate to 3-phosphoglyceric acid, which is the 6th step, okay, which is the 6th step of glycolysis, wherein NAD plus will remove the electrons and protons from the substrate. It will remove the electrons from the substrate, forming out what? NADH plus H plus. Okay, this forms out to be what beta? NADH plus H plus. As I have already discussed with you, in the cytoplasm what happens if the cell is conducting anaerobic respiration, then this end products will be the starters for anaerobic respiration, wherein in cytoplasm once NADH plus H plus is released, this NADH will be utilized, either pyruvic acid turning to lactic acid or pyruvic acid turning to ethanol. This NADH is utilized and this is getting converted to what beta? NAD plus. This gets converted to what? NAD plus. So, remember that is why in aerobic respiration the complete energy, total energy in anaerobic respiration will be just 2 ATP. That 2 ATP is also what? Net ATP of glycolysis. NADH energy will not be released because the NADH which is released into the cytoplasm, that NADH is utilized, be it in lactic acid fermentation or alcoholic fermentation. Clear right now beta? Now you have to choose it correctly. A is what? A is NAD plus to NADH plus H plus. Okay, B and C will be what? B will be NADH, it is converted to what? NAD plus. Here also it is converted to what? NAD plus. As such, option C will be the correct answer here. Right now, ma? Remember, NAD plus will remove the electrons, protons from the substrate. As such, it is giving or it is reducing. NAD plus is reducing to what? NADH. If in the same cell, same cytoplasm, anaerobic respiration is conducted, then this NADH is utilized by the pyruvic acid and it gets converted to NAD plus. This is utilized again by the glycolysis. So, this completely continues inside the cell of anaerobic respiration. Clear right now, ma? So, here the option is option C. Okay. Good afternoon, Rashak. Yes. Identify, look into the questionnaire beta. Identify A, B and C in the given reactions of lactic acid fermentation. So, the question is about what? Lactic acid fermentation, and select the correct option he is asking you. Pyruvic acid along with what? Lactic acid fermentation, just now I have told you, beta, along with NADH, A will be what? NADH, that gets converted into what? If it is lactic acid fermentation, there is no way carbon dioxide is being released. So, here B or C, it will be what? Lactic acid or lactate and NAD plus. Fine. So, in the lactic acid fermentation, what is A, B and C he has asked in the question. So, A definitely it will be what? Pyruvic acid along with NADH catalyzed by the enzyme lactate dehydrogenase, and what are the cofactors over here beta? Zinc will be the cofactor, and at the end what products are formed? Lactate and NAD plus from NADH. So, here A is what? NADH. So, you can consider these two. B will be lactic acid and C is NAD plus. Carbon dioxide will be evolved only in the alcoholic fermentation, not in the lactic acid fermentation. So, here will be the option B is the correct answer. Look into this question beta again, assertion and reason type of question, wherein assertion in alcoholic fermentation the hexose molecule is converted to glucose and fructose. Wrong, na? In alcoholic fermentation what will happen? Primarily it starts with what beta? Pyruvic acid. Pyruvic acid with the enzyme pyruvate decarboxylase, with the enzyme pyruvate decarboxylase, it gets converted to what? Acetaldehyde and carbon dioxide. Okay. When to this acetaldehyde, which is catalyzed by the enzyme alcoholic dehydrogenase or alcohol or ethanol dehydrogenase, also we can say it as dehydrogenase. Lastly, what is formed ma? Ethanol and carbon dioxide, ethanol plus carbon dioxide. Fine ma. So, here if you see assertion and reason type of question, in alcoholic fermentation hexose is not converted to glucose or fructose. In alcoholic fermentation pyruvic acid is converted to ethanol and carbon dioxide. So, this will be wrong. In reason if you see alcoholic fermentation is anaerobic respiration brought about by the enzyme ATP synthase. This is wrong. What is the enzymes that are catalyzing this reaction? Pyruvate decarboxylase and alcoholic dehydrogenase. It is not ATP synthase. So, here what we have to choose ma? Both assertion and reason will be wrong. Option D should be our answer. Next question. Any doubt anywhere you can ask me ma. Students, just go through the questions. Most important session because I have chosen only difficult questions today. Okay. There are three major pathways in which different cells handle pyruvic acid produced by what? Glycolysis. These are what? Okay. So, here pyruvic acid, if it is going towards the aerobic respiration pathway, pyruvic acid if it is going for aerobic respiration, this pyruvic acid will be directly landed to where ma? Into the mitochondrial matrix. This directly lands to mitochondrial matrix conducting link reaction and tricarboxylic acid cycle or Krebs cycle inside the mitochondrial matrix. Do remember, link reaction and Krebs cycle both will happen in mitochondrial matrix. And if you have to see anaerobic respiration, so in anaerobic respiration pyruvic acid can either conduct lactic acid fermentation or it can conduct alcoholic fermentation. Clear ma? So, what are the three pathways where pyruvic acid can choose after glycolysis? It can be fermentation, aerobic respiration, alcoholic fermentation. So, this will be the answer ma. Option C will be the answer because here everything is covered. What is that? Alcoholic fermentation it can continue, lactic acid fermentation it can continue, or aerobic respiration. Aerobic respiration involves all the pathways. What? Link reaction or pyruvate decarboxylation and TCA cycle and ETS. So, it involves all the three. What are they ma? Link reaction, TCA cycle, and one more thing, last step of aerobic respiration is electron transport chain. All the three will be involved. Clear? So, here the right answer is option C. Correct now? Next question, next question you see this one, which is asking you about TCA cycle, which is very important. Look into that properly. Pyruvate to acetyl coenzyme A, first labelings he has asked. So, mark out the labelings properly. Here B is given and here D is given, A, C, E. Clear, right? So, this how you have to look into. Of course, let us start it step by step. First B, if you have to see the immediate step, once the acetyl CoA, which is 2 carbon compound, the primary acceptor of acetyl CoA in TCA cycle is what? Oxaloacetic acid. So, here E will be oxaloacetic acid. OAA, which is 4 carbon compound along with 2 carbon compound, this will be forming what? Citric acid. Citric acid. So, B is what here beta? It is citric acid. And then after citric acid formation, one decarboxylation is happening, carbon dioxide is released and NADH is released. So, D will definitely be after decarboxylation and NADH energy release, it is alpha ketoglutaric acid. After this, one more decarboxylation, it forms acetyl coenzyme A. Acetyl coenzyme A undergoes substrate level phosphorylation forming GTP, and here A will be what? Succinate, succinate or succinic acid. After that, again the succinic acid will be turning towards what beta? Fumarate, releasing out FADH energy, fumarate and malic acid at the end. So, here C will be what? Fumarate or malic acid, anything will be correct only, malic acid. Fine. So, here you see the options carefully. Malic acid will lastly convert to OAA. Look into the options carefully First, B is what? Citric acid, and then D will be what? Alpha ketoglutaric acid. Yes. And next A is succinic acid or succinate. C will be what? NADH. Before the NADH release, it is of course fumarate or malate, anything is fine. So, C will be fumarate or malate, and E is of course oxaloacetic acid. So, here the right answer is what Option C. Right answer will be option C. Clear, right? Next question you look into beta. This one you see. Yeah. In TCA cycle, the question is again about TCA cycle, wherein the formation of isocitrate is followed by 2 successive steps of decarboxylation. So, here what happens? Isocitrate which is 6 carbon, right? Isocitrate which is 6 carbon, it will lose 1 carbon dioxide undergoing what reaction? Decarboxylation, and at the same time it will release out NADH energy also, 1 molecule of NADH, and it gets converted to 5 carbon compound, that is alpha ketoglutaric acid, which is 5 carbon. Okay. This 5 carbon compound again undergoing decarboxylation, that is losing 1 carbon dioxide, at the same time 1 molecule of NADH again it will be losing, right and it forms 4 carbon what? Succinyl CoA, 4 carbon succinyl CoA is formed, succinyl coenzyme A. So, here you look into what is the correct answer. Of course, it involves 2 successive steps involving alpha ketoglutaric acid and succinic acid. Nowhere citric acid is involved, malic acid is involved, succinic acid no. These both are 4 carbon only, both of them. So, 2 steps it will be involving from citric isocitrate, which is 6 carbon. First is it turns to 5 carbon compound, then it is turning to what? 4 carbon succinyl CoA. At each step 1 carbon dioxide is released and 1 NADH will be released. Clear, right Next question you see. Next question. Yeah. Find the values of W, X, Y and Z in the given equation. So, in this how you have to identify when such questions are asked. First look into the major step, or first look into the major substrate. Here what will be the substrate, ma? The substrate is pyruvic acid. Pyruvic acid is the substrate, and at the end what it is releasing? Just energy, that is NADH, carbon dioxide and FADH2. It means that it is conducting two steps, right? That is link reaction and tricarboxylic acid cycle. Both of them are operated where? Both of them are operated in mitochondrial matrix. Mitochondrial matrix, because the pyruvic acid after glycolysis inside the cytoplasm, it directly enters into the mitochondrial matrix, and then here first pyruvic acid, one molecule of pyruvic acid will undergo one decarboxylation step by utilizing how much? 1 NADH, by utilizing 1 NADH, sorry NAD plus, it will be forming what? Acetyl CoA and 1 NADH is released out and one molecule of carbon dioxide will be released out. This is about what? Link reaction, where it is forming acetyl CoA and one molecule of NADH and one molecule of carbon dioxide. Second, third and fourth if you see, okay, that is link reaction, that is what? TCA cycle. In TCA cycle what happens? At 3 locations 3 NADH will be released. Yes or no? At 3 locations 3 NADH will be released. At one place, that is from succinic acid to fumaric acid, when it is, the step is succinic acid to fumaric acid, at that step 1 FADH2 is released. 1 FADH2 is released, and totally in link reaction 1 carbon dioxide is being released, and in Krebs cycle 2 carbon dioxides will be released. As such, when you combine both link reaction and Krebs cycle, totally 3 carbon dioxides will be released. So here you look into the question properly, pyruvic acid plus 4 NADH, can I write that beta? 4 NADH, because 1 NADH is in the link reaction and 3 NAD plus, sorry 1 NAD plus in the link reaction it is utilized, and 3 NAD plus will be utilized in what? Krebs cycle. As such, 4 NAD plus, 4 NAD plus, and how much FAD plus? Z will be 1 FAD plus only, because only 1 FADH2 will be released in the TCA cycle. And how much water molecules will be released out? That is of course 2. In the citric acid cycle, only 1 water molecule will be released, and then it is hydrated. Hydration, dehydration, both the steps will happen at the beginning of TCA cycle. So XO also will be what? 1 only. Then ADP plus inorganic phosphate. After that, by how much carbon dioxide will be released combiningly, link reaction and TCA cycle, it is 3 carbon dioxide, okay, 3 CO2, and totally as 4 NAD plus will be utilized, 4 NADH will be released out, and how much H plus will be released out? Of course it is 4 again, okay. How much H plus? W, H plus plus Z, again FADH2 will be 1 only, W is 4, fine. 4 NAD plus will give out 4 NADH plus H plus, okay. 1 FAD plus will give out 1 FADH2, and 1 water molecule will be released out, 3 carbon dioxides on total, understood, combining link reaction and TCA cycle. As such, you have to code it beta. When such combined reactions are given, summary reaction of link, summary reaction of what? Summary reaction of link reaction and TCA cycle. When that summary reaction is given, then you have to follow up in this way, looking up to the substrate and the products that are formed. You can identify what type of reaction is taking place. Now you look into the question properly. As such, what are formed? 4 NADH. So W will be 4 over here, and Z will be 1, W will be 4, Z will be 1, X will be 2 water molecules of course, and Y is what? 3 carbon dioxides. As 2 water molecules are evaporated in TCA cycle, where both hydration and dehydration are happening. So here the right answer will be option A. Next you look into this question. Here the question is about just end products beta, which is very simple and easy when compared to previous questions, but then the level of the question is difficulty level only. Look into here properly. Glucose after glycolysis, this forms pyruvic acid. Pyruvic acid. Glucose after glycolysis, it forms pyruvic acid. Pyruvic acid after Krebs cycle, what will be released? Carbon dioxide, energy, water molecules will be released. Carbon dioxide, water, and how much total after Krebs cycle? In Krebs cycle only 3 NADH and 1 FADH2 and 1 ATP will be released. Clear, right? Amongst these products, whichever are there, we can choose. Fermentation. In bacteria, that is lactic acid fermentation. So, at the end only lactate is formed. Clear, right? Yeast. Yeast will undergo what fermentation, ma? Alcoholic fermentation. It will undergo alcoholic fermentation, forming what? Ethanol and carbon dioxide. Fine. So, look into it clearly. So, here A is what? Pyruvic acid. B will be carbon dioxide and water. C cannot be ethanol. So, we cannot choose this option. And look at D beta. A is what? Pyruvic acid. B will be carbon dioxide plus water. C can be lactic acid or lactate. D will be ethanol and carbon dioxide. So, here the option will be option D. Clear, right? Next question let us look into. Yeah. All enzymes of TCA cycle are located in the mitochondrial matrix except one, which is located in inner mitochondrial membrane in eukaryotes and in cytosol of prokaryotes. This enzyme is, we know, just now we have learned that there is one substrate level phosphorylation, one SLP that is happening in TCA cycle or Krebs cycle, right ma? So, this SLP is happening where? From succinyl coenzyme A, succinyl CoA to succinic acid. This is catalyzed by what enzyme? Succinate dehydrogenase enzyme. Here either one molecule of GTP might be or ATP might be, will be released. This is a substrate level phosphorylation and the enzyme is what? Succinate dehydrogenase. Okay. This succinate dehydrogenase is popularly known or it is well known as complex 2 in the electron transport system. This complex 2 is located in the inner mitochondrial membrane. Okay. Succinate dehydrogenase enzyme or complex is located where? In the inner mitochondrial membrane. Very important ma. So, here the answer is what? Option C will be our answer. Clear, right? Next question. This you see, the question is directly from the diagram of electron transport system. Yes. So, here you see ma, A, here where are NADH, FADH2, the energy rich molecules are formed, that is in mitochondrial matrix, right, because TCA is happening in the mitochondrial matrix. So, that energy rich molecules are formed inside the mitochondrial matrix. So, A will be what? Mitochondrial matrix. So, here inside the mitochondrial matrix, what is happening? First NADH will be dehydrogenated to what? NAD plus plus H plus, and that protons are sent to intermembrane space. Okay. So, here B will be what? Intermembrane space. Intermembrane space. Clear, right? So, this B will be intermembrane space. Now, look into C. C is what? Our complex 3. Okay, this is complex 1 NADH dehydrogenase and this is complex 2, that is succinate dehydrogenase. C will be what? Cytochrome BC complex. So, here 3, here this is what ma? Cytochrome B. Fine. And D is what? Cytochrome C1. This is E is what? Cytochrome C. As such, F will be what? Cytochrome A, because this is cytochrome A3 complex, last complex, complex 4. So, here you see the options correctly. A is mitochondrial matrix, B is intermembrane space. So, you can opt this and this, and then C will be what? Cytochrome B. Okay, both of them are correct. D is what? Cytochrome C1. Okay, D is cytochrome C1 and E is cytochrome CF, that is over this in the complex 4, cytochrome A3 complex. So, this is what? Cytochrome A. Fine. And one more point to make you clear that in all this complex 1, 2 and 3, the protein molecule or carrier which is present is ferrous sulfur complex, whereas in complex 4 you can see copper ions. Okay. So, that you make a note of it. So, here this is about the labeling of ETS. One more question I have asked based on the labeling of ETS. Yeah, this you see ma. One more nice question about ETS. Identify P, Q, R, S and T in the given diagram of mitochondrial electron transport chain. So, here again Q will be what? Mitochondrial matrix. Always do remember when the labelings are given, the flow of protons will be towards the inner membrane space. So, if the intermembrane space. So, if the arrow is like this, then this space is what? Intermembrane space. Intermembrane space. So, here Q is mitochondrial matrix and P is intermembrane space, and R here will be, as it is complex 1, R here will be what? NADH. Fine. S will be what? NAD plus plus H plus. Fine. And T over here is releasing you what? Protons. Okay. It will be 4 protons. Clear, right? So, this is about T. Look into the options properly. Q will be what? Q is mitochondrial matrix. So, you can't opt this. Yes, this is correct. And P is what? Intermembrane space. R can be NADH plus H plus. S over here will be what? NAD plus, and T will be 4 protons. It is not 2, it is 4 protons. So, here the option is B. B will be the right answer. So, with this today's questions will be done ma. Tomorrow we will look into the most balanced sheet of the questions, all the bioenergetics of glycolysis, Krebs cycle, link reaction, anaerobic respiration, complete energetics and numerical part, along with few questions involved from ETS we will be solving tomorrow. So, do not miss tomorrow's class at all, and I would like to end the session by giving you one more time the complete introduction about NEET World's special programs. Just a second, this slide is loading. Yes. So, we have 3 special programs of NEET World, that is for 11th students, 12th and for repeaters, under the names of 2 steps to medicine, 1 step to medicine and power step to medicine. All the 3 programs will be with the same minimal fee of 3333 per month. To know more about these courses, you can call us to the given number, or you can directly visit our branch which is in Narayanguda, Hyderabad. And lastly, I would like to say this to all the students who are learning in the public platform or who are preparing for self-study for your NEET 2026. To you people, you know, this NEET World is with you. For free of cost, you can log into our Telegram group, wherein you will get the complete access on daily quizzes of biology, chemistry, physics and mathematics, and then daily live sessions will be updated in that, free resources will be provided, one-on-one doubt solving sessions with faculties are given. And then to log into this Telegram group, you can scan this QR code or call us to the given number, or you can directly visit our official website and you can download the Telegram group. So, with this I will sum up today's class and we will meet tomorrow with the questionnaire session again. Okay, thank you.